Question is simple. They just asked about the activity of catalase on the three different test tubes, CR and H, and then they find the height of the form that is produced. Okay, what is this form made up of basically? What is this form? Okay, w- n- okay, okay. Wait. Read the first line. Read the first line again. Yes, so this one is oxygen gas. Okay, so let's say the H has okay, which test tube has the highest activity of enzyme? H. Exactly, because more volume of hydrogen is produced, and in C it's least. Okay, prepare a table for the result of investigation. Measure the maximum height of the form in millimeter in each of the test tube and record these measurements in your table. So we need to take out, yes. Okay, okay. Because this is an error. They would ask you further what is the error. Okay, no, we will take the highest one, like height. Which one is whatever is at the top, we'll calculate it from the top. Okay. Okay, so we'll make a table and there would be two columns. On one of the column we would write test tubes. So under test tubes we have three rows, 1, 2 and 3, and the other column would be of height of the form, and then we write the height of form. And what would be unit for height of form? Millimeter. Okay, let me check if I can add something over here. No, it's saying you can only. Okay. So it would be like this, height of the form in millimeters, and then in front of this we would write this cut. C is 8 millimeter for me, so, and R is 15 millimeter, and H1 is 27 millimeter. Okay. Okay, the next one is state a conclusion of this result. Exactly. Can we write higher the temperature? Good. Okay. As the temperature increases, don't write short forms like me. Just write the full one, okay? The activity of catalase enzyme also increases. What is the independent variable and what is dependent variable? And this one is usually given in the first line. Temperature. So exactly. Okay. How is it visible? That is by a form. So independent one is the temperature, and dependent one is height of the form. Okay, next one is explain why test tube C and test tube H were kept in water bath for 5 minutes in step 6. So why 6 minutes? Hmm. It can be. What else? So that each part of that test tube would have the equal temperature. So we can write so that the temperature of contents, of all contents, uh, contents become same. Okay, there is another word we use for it, to equilibrate the, like it should be at equilibrium, let's say. Okay, the student suggests repeating the investigation twice. Explain how would this give you more confidence in this result. So yes, we do it to get a reliable result. Okay. Okay, so yes, so good, good, to excluding the what was word, anomalous result, anomalous result by identifying them. After identifying them is better. After identifying them. Okay. Potato extract can be prepared by cutting potatoes in two pieces and then grinding it with mortar and pestle and some water. Now you have to give a safety precaution to prevent or reduce any hazard. Now you are cutting and then you are grinding. So there can be two hazards. Mention any one. Good. Okay. And if you're cutting, good, good. Cut it on a hard surface or a stable surface. Okay. Hard surface or cut it away from the body. That is also correct. Okay. Next one is another student did the same experiment and measured the height of the form in one of the test tube as 29 millimeter and the diameter of the test tube was 25 millimeter. Calculate the volume of form and give your answer in cubic centimeter. So first of all, we need to convert them into cubic centimeter. How can we do that? Convert. We just divide it exactly. So height is, let's say height is 2.9 centimeter and diameter is, or width is 2.5 centimeter. So what would be volume? That is pi. Correct. That would be 1.25. Okay. Okay. Do it then. 2.9, yes. Okay, it's a volume. Keep this thing in mind. Hmm. How you are multiplying it? What is the formula that you are applying? Let's say it's a volume of a cube, a cuboid, cube or cuboid. How do you calculate that? Okay, there is one what do we say? One term in this which would be square. Hmm. Hmm. Now calculated. Why they have given this? I'm not getting it. Yes, it is 14.2, 14.2 cubic centimeters. So it would be pi, height, and r square. Okay. Suggest a more accurate method of measuring volume of oxygen gas produced. Now that was an error that you figured out at the start, that they are not at the same level. So what is another way? Yes, use a gas syringe. Okay. Next one is iodine solution was used to test a sample of potato for starch. State the result of positive test for starch. Only the positive test result. Blue black color. And what is the reagent we use for the identification of vitamin C? DCPIP. Photosynthesis also produces oxygen gas. Plan an investigation to check the effect of light intensity on the rate of photosynthesis in aquatic plant. This is the simplest question so far. Okay. So the very first thing, you need to give the variables, and then need to give the method, and then the constants, and then the repetition. Okay, start now. क्या हो गया मेरी बिल्ली को? बहुत थकी हुई है. Okay, okay, tell me. Okay, tell me. Okay, so 2 to 3 different light intensities would be investigated, and how would you calculate it? Okay, wait. You have given me only the independent variable. Na? 2 to 3 different light intensity would be investigated for what? What is the dependent variable? And the volume of oxygen was measured for them. Okay, now give me the method. Hmm. Hmm. Exactly. Exactly. Correct. Correct. Then. Hmm. Okay. Correct. 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 Okay. Correct. So the method is, uh, the light intensity would be changed by changing the distance of lamp from the test tube. The gas syringe would be attached at the mouth of test tube to collect oxygen gas, right? Okay. And what was the third thing that you mentioned? There was something else. And okay. It was related to something same. It like time would be, and the same time after, and the oxygen collected was measured after a Constant time, right? Okay, now give me the constant variables. The plant would be of same size. What do you mean by same size? Same species. What do you mean by same size? Same number of leaves, same species. Okay. What else should be same? The temperature should be kept constant. Sodium hydrogen carbonate would be kept constant. Okay. The wavelength of light would be constant. That we have to use the same light. Okay. Exactly. Okay, there was one more thing I was about to tell, that in such experiments where we are checking the light intensity, we usually put that test tubes in which that aquatic plant is present in a beaker, because we are s- sometimes when we use the bulbs, so that could heat up the one side of the test tube and not the other. So it's better to keep that in water bath, so that the temperature remains same for the like whole beaker, not from one side only but for the whole beaker. Okay. And we need to, exactly, we can. Okay, so what is left? The last thing. 2 to 3 times to get the reliable result. Okay. Um. Is there any need to? Yes. You can also say when you give a conclusion that a plot can be. Sorry, we can plot a graph. Sorry, we can plot a graph. I was about to say plot can be grafted. A graph can be plotted between the light intensity and the volume of oxygen collected, right? Okay. The next question. Yes. Okay, let me check it. So, okay, a tail is drawn, that is correct. The overall shape is good, the tip is good. So, is it a 4 mark? Let me check. Where is the paper? Then all the requirements are done. This was an easy diagram. Okay, the question is calculate the magnification. Now, since you have your own diagram, you can. Okay, I'm giving you the length for this PQ. The length for this PQ is for me it's 82 millimeter. How, what is the height of your drawing? Okay, so it would be 114. Okay, now you have to calculate the actual length. Okay, what's the magnification given? The magnification is 3.4. Okay. No. Wait, we need, okay, no, we don't need for diagram. We need it from that line PQ. Line PQ. Line PQ. Okay. So that would be 25. Okay, correct. Next one. Okay, this is a photograph of a corn from Caucasian oak and from a Turkey oak. So, what is the difference between two? Visible, like visible. It's written cupule. It's written. Okay, correct. Okay, okay, lighter color. Okay, two differences are enough. Or moreover, we can talk about the tip. The tip for this oak is more pointed as compared to the other one. Okay. Different species of oak tree have acorn on different masses. Okay. Some scientists investigated if the mass of acorn affected the proportion of acorn that germinate in three different species of oak tree. They took almost 100 acorn for each of the three species, and then they take the mass and then they place them in a dark room at 25 degrees Celsius and water them for 48 days. Why they were kept in dark? Why were they kept in dark? It's written in the third point that they were kept in the dark during germination. Unite, exactly. Okay, state two factors that were kept constant. Were means that are given in this investigation. Hmm. Okay, temperature and species. Okay, next one is a table is given and you have to draw a graph. Okay, why a large number of acorn were measured? Uh, no, no. No, no, no. We have done such question when we calculate like a diagram was given in which half flower was outside, and then we have to calculate the number of flowers in the region. At that time, we talked about this that they how do we calculate or get a good? Why do we use like so many things for accurate reading or for a representative data? Okay, okay. Next question is related to percentage. Calculate the percentage water content of species B. This is not given. So it would be simple. The initial mass which has water and the dried mass. So it would be, and you have water plus acorn and only acorn. Just subtract and then find the percentage for that. Okay. Yes, we do. We need to. Yes. 62.2. That is correct. So initial minus whatever is this, whatever minus sign is this divided by 1.96 multiplied by 100. Okay, working is not required. State the name of reagent used to test for protein. Reagent. So, okay, we are done with this paper. Let's move to another paper.